Welcome to the latest Novogene webinar, A Beginner's Guide to DNA Sequencing, Recent Advance. Today's webinar discusses the recent advance in the sequencing of the human genome and the other animal, plant, and microbial species. All our previous webinars are available to view on demand on our website. A bit of housekeeping before we start. The presentation will run for around 40 minutes with 15 minutes for questions and answers at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. We'll try and prioritize the most popular questions. If we don't have time to answer your question, we'll come back to you via email after the webinar. Please don't use the raise hand functionality. Just send any questions via the Q&A or message us directly if you have a technical issue. We will share the slide deck and a recording of the webinar via email. You can expect this on Friday. If you are studying and need a certificate of attendance, there will be a link in the follow-up email that will display the certificate. You can print from your browser. Finally, there will be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this, as helps us to plan future webinar topics. I would now like to introduce your host of the webinar, our scientific marketing writer, Aoife. Aoife, over to you.
Thank you, Miranda. Morning, everybody, and thank you for joining our Beginner's Guide webinar on DNA sequencing and its recent advances. My name is Aoife. As Miranda said, I am Novogene's scientific writer and your host for today's webinar. Shortly, I will pass over to Reid, who will talk you through current methodologies used for DNA sequencing, sample requirements for various applications, and some commonly used data analysis approaches. Before I do that, I'm going to speak a little bit about Novogene, especially for anyone who's joining us for the first time, and tell you about what we can offer you to assist with your next generation sequencing requirements in your research. So Novogene was founded in 2011 and has been rapidly growing ever since. Our company mission is to advance genomics and improve life. And as you can see here, our services cover a huge range of applications with real life implications, from complex disease research and oncology to food and water safety. Our European Sequencing Centre is based in the Cambridge Science Park in the UK. Here we have our state-of-the-art lab equipped with the latest Illumina NovaSeq 6000 sequencing machines. We have always provided fast turnaround times and high quality data, but that has improved even further recently with the installation of Falcon 2, which is our new intelligent sequencing platform. This has increased our monthly capacity by 200% compared to manual steps. And we also have our supercomputing clusters, so we can offer bioinformatics services and store your data in the UK. However, at our heart, we are a global company. We can offer services across Asia and the US, and of course, Europe. Falcon 2 has now been deployed across a number of our labs, standardizing our services globally. With almost 2,000 employees and serving over 5,600 customers in world-class universities, biotech companies, and hospitals, we can provide sequencing for the smallest or the largest of projects. We provide a full range of next-generation sequencing services, such as whole genome sequencing, whole exome sequencing, various types of RNA sequencing, metagenomic services, and epigenomic services. These can be applied to a wide range of research topics, from cancer and rare disease, as I mentioned, to microbial cell factories and gut health, and to agriculture and animal health. The focus of today's webinar is the sequencing of DNA and recent technological advances in that field. We use DNA sequencing methods across the majority of our services. We use it for whole genome sequencing of humans, plants, animals, and microbes, for more targeted approaches, such as whole exome sequencing, our various metagenomic sequencing services, and also for our ChIP-seq and whole genome bisulfite sequencing services. Many other companies offer these services too. The NGS market is quite crowded, so why Novogene? There are a lot of good reasons why you should trust Novogene with your project. As I mentioned, we have had a really exciting development recently in the installation of the Novogene Falcon 2, our new intelligent NGS delivery platform. Also, as I mentioned, it's now been deployed in our Chinese, US, and Cambridge labs, standardizing our global service. This new technology was developed in-house by Novogene and has already massively increased the capacity of the Cambridge Sequencing Centre. It will also increase the quality of the data produced as it is entirely automated and will remove any possibilities of human error. With this new addition, we are now able to offer our fastest ever turnaround time for RNA and whole genome sequencing, just 10 working days from sample arrival to data release. Falcon 2 really highlights the innovation that we strive for here at Novogene in an effort to keep pushing the frontiers of genomic research. The benefits of our in-house innovation are clear in our record in the scientific community, which we are really proud of. We have been cited in over 790 research papers with a total impact factor of over 6,120. We are also driving the NGS industry forward with over 37 NGS-related patents. Our technology is helping to make the NGS process more efficient and effective, all the while improving the quality of the data that we deliver. The Novogene community keeps on growing. Our expert sales team speaks over 10 languages and is located across Europe, ready to assist in your next project. If you're not ready to send us samples just yet, you can still benefit from our educational outreach to learn how NGS can work for your project. Today actually marks the first day of the new leg of our European Roadshow tour, where members of our sales and product teams go out and deliver seminars about the applications of NGS in DNA and RNA research. Miriam is currently delivering a seminar in Barcelona, and other dates in Spain and Portugal have also been announced. So keep an eye out on our social media for future seminar announcements, and hopefully we'll be visiting somewhere near you soon. I'd like to just quickly introduce our sales team. So they are, as I said, based all across Europe. So we have the UK and Ireland team, our DACH region team, and our Benelux region team. We've also got our Southern European team, which has expanded quite significantly recently. We've got our Nordic and Eastern European teams. So if you would like to speak to your local sales representative about an upcoming NGS project, please contact us via our website and you will hear back from your local salesperson. So with that, I'd like to thank you for your attention, and I will hand over to Reid. Reid holds a PhD in molecular epigenetics and is one of our product marketing managers. With his expertise, he's going to guide you through the basics of DNA sequencing and some recent advances in the field. So over to you, Reid.
Thank you, Eva, for the introduction, and thank you everyone for joining us today. I just need 1 second to share my screen. Okay, perfect. So thank you again for joining us today. And before I start with my presentation, I would like to give you an overview of today's talk. In the first part, I'm going to talk about DNA sequencing in general, along with the current DNA sequencing technologies that are currently available on the market. After that, I'm going to highlight a few applications of DNA sequencing that can be used for different living organisms. And after that, I'm going to show you the general DNA sequencing workflow. Towards the end of my presentation, I'm going to conclude with a summary and an outline. So to start with, we know that DNA sequencing by definition is the process of determining the nucleic acid sequence. In other words, we are interested in determining the order of nucleotides within the DNA molecule. In fact, the sequence of the nucleotides that we know as A, T, C, and G is responsible for encoding the biological information that cells use in order to develop and perform their function. Understanding or unraveling the DNA sequence is crucial and fundamental in order to understand the function of genes and the genome overall. Currently, there are different methods available for DNA sequencing, where each one has its own characteristics in terms of advantages and disadvantages. If we take a look at the evolution of the different sequencing tools, we can see that the first generation is mainly represented by Sanger sequencing. Using Sanger sequencing, we are able to sequence DNA fragments ranging in size from 500 up to 1,000 base pairs. After the first generation came the second generation sequencing tools, and this is mainly represented by Using the Illumina platform for DNA sequencing, we are able to sequence DNA fragments ranging from 50 up to 500 base pairs. However, in the last couple of years, there have been a lot of developments and advancements into the field of third-generation sequencing tools. These tools are mainly represented by two main platforms, Oxford Nanopore Technology and Pacific Biosciences, or PacBio. Using both of these platforms, we are now able for the very first time ever to sequence DNA fragments that are tens of kbs in size on average. Now, focusing on the different sequencing principles of the different platforms, we can start with Sanger sequencing. The workflow for Sanger sequencing starts with the annealing of the sequencing primer and the chain extension reaction, during which fluorescently labeled nucleotides will be incorporated into the DNA. The product of this step will be a fluorescently labeled DNA sample that will undergo capillary gel electrophoresis. Eventually, the machine is going to detect and quantify the fluorescent signal and assign a DNA sequence accordingly. One of the main advantages of performing DNA sequencing with Sanger is expenses. It is considered cheap and cost-effective for a low target number. Additionally, it has very well-established workflows and pretty simple data analysis pipelines. However, sequencing with Sanger can have some disadvantages, including low sensitivity and scalability, and more importantly, restrictions or limitations in terms of sample input requirements. Now, moving on to the second generation sequencing tools, in particularly Illumina, we know that the first step in the workflow is library preparation. During this step, the DNA will be fragmented into smaller pieces and later ligated to sequencing adapters with the aim of preparing a DNA library. Afterwards, the DNA library is loaded into the machine, where the DNA fragments are going to hybridize to the flow cell. Following several bridge amplification cycles, the DNA will be in the form of amplified clusters on the flow cell. During sequencing, several fluorescently labeled nucleotides will be incorporated through various sequencing cycles, and meanwhile, the machine will be measuring the fluorescent signal and converting that into a DNA sequence. Sequencing using Illumina has a lot of advantages, including increased sensitivity, more data that can be generated with the same DNA amount, and more importantly, high throughput. High throughput, and that is because the sequencing machine is able to provide us with millions of sequencing reactions in parallel. However, sequencing with Illumina is considered to be expensive for low target number. It can be time-consuming, and one of the major limitations or restrictions is the limited short reads, where we can sequence only up to 500 base pair. Now, moving on into the third generation sequencing tools and starting with PacBio, the first step in the workflow is the ligation of the smart bell adapters to the double-stranded DNA. After that, the DNA library will be loaded into the machine, where a DNA polymerase that is fixed at the bottom of the well will start sequencing the DNA fragment using several fluorescently labeled dNTPs. During the synthesis and the polymerase activity, the machine will be measuring the fluorescence and assigning a nucleotide sequence that we can actually obtain and analyze. One of the main advantages of sequencing with PacBio is the possibility to choose between two different sequencing modes. The first one is called continuous long reads, or CLR in short, that is more compatible with large insert sizes. If we decide to use the CLR mode for sequencing, what will happen within the instrument is that the DNA polymerase will sequence each DNA fragment only once. On the other hand, the second sequencing mode is called circular consensus sequencing, or CCS in short, and that is more compatible with small insert sizes. Sequencing with the CCS mode would enable the DNA polymerase to sequence the same DNA fragment multiple times. As you can expect, because we are sequencing the same DNA fragment multiple times, any base errors that might have happened during the first or the second measurements will eventually be corrected. This, in the end, will provide us with highly accurate reads called high-fidelity reads or hi-fi. In addition to these two sequencing modes, among the other advantages of sequencing with PacBio include the possibility to sequence long reads, high accuracy with the CCS mode, and for the first time, a direct detection of epigenetic modifications. On the other hand, sequencing with PacBio is considered to be expensive. It requires large amounts of starting material, and more importantly, there is still a high error rate with the CLR. And finally, discussing Oxford nanopore technology, the sequencing principle here is quite different. The technology itself relies on a nanopore protein that is fixed on a membrane. During sequencing, a single strand of the DNA molecule will pass through the nanopore from one side of the membrane to the other. With every nucleotide within the DNA strand crossing through the nanopore, it will be accompanied by a change in the ionic current. The instrument will rely on the change or the drop in the ionic current in order to assign a nucleotide identity. And this is how we are actually able to do the DNA sequencing using Oxford nanopore. The advantages of using Oxford nanopore for DNA sequencing include long reads, even longer than PacBio, and for the very first time, direct sequencing of the RNA, allowing us to omit the cDNA generation step. In parallel, we are also able to detect RNA modifications. However, sequencing with Oxford nanopore is considered to be expensive. The instrument has high overall error rate in general, and large amounts of starting material are needed. Now that we have talked about DNA sequencing and the different DNA sequencing technologies that are currently available on the market, in the next couple of slides, I'm going to focus on the different applications of DNA sequencing. In this diagram here, you can see a summary of the most commonly used DNA sequencing applications, whether we are interested in investigating the genome or the epigenome. So for those of you that are interested in using DNA sequencing to investigate the genome, we can perform whole genome sequencing with two different approaches, either de novo or resequencing. In addition to whole genome sequencing, we can also perform whole exome sequencing, target region sequencing, and metagenome sequencing for microbial research with one of two approaches, either amplicon-based or shotgun-based. In addition to the genome, DNA sequencing can be used to investigate the epigenome through different applications such as bisulfite sequencing, ChIP-seq, RIP-seq, ATAC-seq, and many more. Starting with whole genome sequencing, we know that whole genome sequencing, or WGS by definition, is a technique that is used in order to sequence the entire genome of an organism at a single time. WGS is very important because it provides us with a deep insight into the DNA sequence of living organisms, including humans, animals, plants, and even microbes. With WGS, we are able to identify and analyze different variants of the genome. One of the main advantages of whole genome sequencing is that it can be done with two different approaches. The first one is called de novo, and our aim with this approach is to assemble a reference genome for a specific living organism or species where the reference genome does not exist. Using this approach, what we will do is the following. The genome will be fragmented into small pieces, and later thousands of sequencing reads will be generated. During data analysis, we assemble the sequencing reads in order to build a reference genome that did not exist before. On the other hand, using the resequencing approach, our aim here is different. What we are trying to do here is to sequence a new individual within a specific species in order to find differences between the new genome that we have sequenced and the reference genome that already exists. With resequencing, we break the genome into small fragments and then later generate millions of sequencing reads. After that, using data analysis pipelines, we actually align the sequencing reads into the reference genome of the organism that already exists, in order to find variations or alterations that might have happened between the individual genome and the reference gene. Now, since we are talking about detecting variants and alterations of DNA sequencing, it is very important to know that variation within the DNA sequence can take one of different forms. It can be as simple as a single nucleotide variant, where a single nucleotide is affected, or it can be a deletion or an insertion, and those are commonly known as indels, and they are considered as indels as long as they affect less than 50 base pairs at once. Any variations that affect more than 50 base pairs at once are considered to be structural variants, and these include tandem duplication, intersperse duplication, inversion, translocation, and copy number variant. DNA variation is actually very important because it contributes to the differences between individuals of the same species. More importantly, depending on the location within the genome, DNA variants can have one of two impacts. Either there is no impact, there is no effect on the organism itself, or we might actually have severe consequences. Among these severe consequences, we might end up with a non-functioning or a truncated protein, and this will eventually give rise to certain diseases. Or there is an impaired function of regulatory elements, and this might affect several genes that are located downstream of these regulatory elements. Or there can be a disruption of the cellular mechanisms and the cellular function. Now, with whole genome sequencing, as I mentioned earlier, we are trying to sequence the whole genome of a living organism. So as you can expect, it can become quite expensive. And this is why there have been certain developments to make whole genome sequencing more affordable. And one of those applications is low-pass whole genome sequencing. You might have heard of low-pass whole genome sequencing as low coverage or shallow whole genome sequencing. The principle of low-pass whole genome sequencing relies on imputation algorithms that are able to provide us with high accuracy of variant call detection at a very low sequencing coverage. Generally speaking, low-pass whole genome sequencing is recommended for at least 20 samples, and the experiment itself has a flexible setup, and this makes it easy to implement for new species or custom populations. Several scientific fields can benefit from low-pass whole genome sequencing, such as those focusing on evolutionary analysis, molecular breeding, pharmacogenomics, and many others. Now, moving into the second DNA sequencing application that I'm going to talk about today, we come to whole exome sequencing, or WES. Whole exome sequencing is a technique that focuses on the sequencing of all the protein coding regions of the genes within the genome. These protein coding regions of the genes within the genome are referred to as the exome. Now, sequencing the exome is very important because even though it represents around 1% of the human genome, it harbors more than 85% of known disease-related variants. So by performing whole exome sequencing, our aim is to identify genetic variants that would alter the protein sequences, especially those that are responsible for Mendelian or polygenic diseases, such as Alzheimer's. One of the main advantages of whole exome sequencing is that it is cost-effective compared to whole genome sequencing, and it can be easily applied in both academic research and clinical diagnostics. For every whole exome sequencing experiment, there are two main steps. The first one is called exome enrichment, where we use commercially available kits, such as those provided by Agilent, Twist, or ITT, in order to enrich for the exome within the sample that we are studying. Following exome enrichment, we proceed with DNA sequencing using Illumina as a sequencing kit. In addition to whole genome sequencing and whole exome sequencing, we can also use target region sequencing to investigate the DNA sequence. Target region sequencing, or TRS, is a technique that focuses on the sequencing of a selected set of genes or regions that we think are involved in a specific disease or phenotype. The advantages of using target region sequencing over whole genome sequencing or whole exome sequencing include the requirement of less sample input, And at the end, we are producing a smaller data set that is easier to analyze and process. Target region sequencing can be performed in two different ways. The first one relies on hybridization capture or target enrichment, while the second one is called amplicon sequencing and relies on the generation of amplicons. The differences between these two methods are as follows. The hybridization capture is usually more suited if we are trying to analyze a larger gene content, more than 50 genes on average. It is more comprehensive and has longer hands-on time than amplicon sequencing. There are several applications and several fields that can benefit from the hybridization capture approach, especially for exome sequencing, genotyping, oncology, and gene discovery. On the other hand, amplicon sequencing is used when we have a smaller gene content analysis, less than 50 genes. It is more affordable and has an easier workflow compared to hybridization capture. Several applications benefit from amplicon sequencing, including genotyping by sequencing, detecting CRISPR editing events, and detecting disease-associated variants. I would like to highlight here that hybridization capture usually relies on pre-developed panels, such as those provided commercially by various companies, including Agilent, Novogene, or Roche. And since now we are talking about the hybridization capture method of target region sequencing, one of the very important aspects of target region sequencing that have been heavily used in the last couple of years is oncology. So we know for a fact that DNA sequencing can provide us with high resolution and a global view of the cancer genome. And this is why targeted oncology panels have been developed in order to deliver a comprehensive profiling test for various cancers. One of those targeted oncology panels is called NovoPM2, and this panel provides us with a profiling test for solid tumors. The panel itself analyzes 484 genes that have been shown to be clinically relevant for the diagnosis and/or the treatment of various solid tumors. Furthermore, the analysis that we obtain by using this panel would provide us with a tailored treatment recommendations for cancer patients. In this figure here, I summarized the three major sequencing applications that I have shown you so far, along with the advantages and the disadvantages of each. So starting with whole genome sequencing, in terms of coverage, we know that we are actually able to sequence the whole genome, including genes and non-coding DNA. With whole exome sequencing, we are able to sequence only the exome, while with target region sequencing, we are focusing the sequencing only on a specific set of genes or genomic regions, and that can be between 10 up to 500 genes or genomic regions. In terms of variants, whole genome sequencing allows us to look at all different variants, including single nucleotide variants, indels, and structural variants. So any type of variant that might happen within the DNA can be detected by whole genome sequencing. However, whole exome sequencing can detect only single nucleotide variants, indels, and copy number variants as long as they take place within the coding region of the genome. And finally, for target region sequencing, we are able to detect single nucleotide variants and indels as long as they occur within the regions that we have covered by sequencing. In terms of costs and depth, whole genome sequencing is considered to be the most expensive option and requires a depth of at least 30x. Whole exome sequencing, in comparison, is considered to be cost-effective and requires a depth of 50 up to 100x, depending on the sample that we are studying. And finally, for target region sequencing, it is considered to be the most cost-effective option among these three and would provide us with a sequencing depth of at least 500x. Now, moving on into microbial research, one major DNA sequencing application that scientists and researchers that are focusing on microbial research can use is metagenomics. Metagenomics, by definition, is the study of microbial communities in their original habitats. And it is very important because it provides us with a comprehensive insight into the interactions within the microbial community. Metagenomics can be performed in one of two approaches. The first one is more targeted, and that is commonly known as amplicon-based metagenomics. Using this approach, what we are going to do is the following. After extracting the DNA from the microbial community, we are going to sequence only a selected set of marker genes that we think are present there within the community. On the other hand, the second approach is more global and is referred to as whole metagenomics or shotgun whole metagenome sequencing. Using this approach, what we will do is that we will sequence everything that is present there within the microbial community. And as you can expect, since we are sequencing everything within the sample, it can get expensive. And this is why shallow shotgun metagenome sequencing was developed. So the principle of shallow shotgun metagenome sequencing is to sequence the microbial communities at a shallow depth compared to deep or standard shotgun metagenome sequencing. The advantages of shallow shotgun metagenome sequencing include high accuracy, so we will get a less biased representation of the microbial community and a higher taxonomic resolution. In addition, it would make the whole expensive cheaper because it is an affordable method to provide compositional and usable sequencing data similar to deep shotgun metagenome sequencing. So in other words, we are actually able to get the same amount of information, but with lower costs and without any compromise on the quality of sequencing. I would like to stress here that shallow shotgun metagenome sequencing is not intended to fully replace deep shotgun metagenome sequencing. And finally, one major field of research that can really benefit from DNA sequencing is epigenetics. Epigenetics is the study of stable phenotypic changes that do not involve alterations in the DNA sequence itself. Epigenetics as a system has two main characteristics. The first one is heritability. In other words, epigenetic information can be faithfully transmitted from the mother cell into the daughter cells during mitosis, or transgenerationally from the parents to their offspring through meiosis. In addition to heritability, the system is reversible. In other words, any epigenetic information can be changed, modified, or even reversed at a later point in time. For those of you that are interested in investigating epigenetics and would like to use DNA sequencing, there are various applications that are available right now. In case you are interested in studying DNA methylation, you can perform whole genome bisulfite sequencing or reduced representation bisulfite sequencing. If you are interested in investigating histone modifications and transcription factors, you can use methods such as ChIP-seq. In order to assess chromatin accessibility, different methods such as ATAC-seq have been developed. And finally, for chromatin structure and the 3D organization of the chromatin within the nucleus can be assessed or investigated using chromatin conformation techniques such as HiC. Now that we have seen some of the major applications of the DNA of DNA sequencing and their principles, in the next couple of slides, I'm just going to show you a quick overview of the DNA sequencing workflow. In short, the DNA sequencing workflow consists of 4 main steps. The first one is sample preparation, where the DNA is extracted from the cells or the tissues or the living organism that you are investigating. After sample preparation, the sequencing libraries are prepared, where specific sequencing adapters are added to the DNA. The final DNA library is then sequenced using the instrument of choice, and after obtaining the sequencing results, we can perform data analysis in order to understand what is happening with the DNA sequence. For any successful DNA sequencing experiment, it is very important to start with good quality DNA. In order to assess the quality of the DNA that we have extracted from the tissues or the samples that we are studying, we can perform an agarose gel in order to see a sharp, high molecular weight DNA on an agarose gel. We definitely want to avoid situations where there is a degradation of the DNA or there is a contamination with either RNA or proteins. In addition to the agarose gel, we can also generate a bioanalyzer or fragment analyzer profile. And our aim here is to see a very clean and sharp peak of high molecular weight DNA without any DNA degradation. In addition to the quality of the DNA, it is also important to have enough DNA that we can use for library preparation. For exact quantification of the DNA, we can use different instruments such as the qubit and the nanotron. In this table here, I added the DNA amounts that are commonly used for the different applications. So if we decide to use the Illumina platform to perform whole genome sequencing, whole exome sequencing, amplicon, or shotgun sequencing, we usually need around 200 up to 300 nanograms of DNA, depending on the origin of the sample. However, if we decide to use the third generation sequencing tools, such as PacBio and Oxford Nanopore, now the sample amount is much higher, and we are talking about something in the range of 5 microgram up to 15 microgram, depending on the application itself. Now that we have extracted the DNA and we made sure that everything is fine in terms of quality and quantity, we can proceed into the next step, which is library preparation. During library preparation, as I mentioned earlier, specific sequencing adapters will be added to the DNA to generate a DNA library that is compatible with the sequencing platform that we want to use. When we get to the sequencing step, it is very important to know how much data do we actually need. In other words, how much sequencing should we do in order to get enough data that we can use for data analysis. In fact, the data requirements that we need for the different applications depend on different parameters. So we always need to take into consideration the research aim, the application that we are using, whether it is whole genome sequencing, whole exome sequencing, metagenomics, or others, the genome size of the living organism or the species that we are studying, along with the error rate of the sequencing instrument, and also it is always helpful to take established guidelines in the literature as a reference. So as you can see, the data requirements really depend on the several parameters, and some research needs to be done for this in order to determine how much data needs to be generated so we can actually have enough data for analysis. When we talk about data and sequencing data in particular, there are two main aspects that we need to take into consideration. The first one is called the depth of coverage, and the second one is called the breadth of coverage. The depth of coverage that is represented here on the y-axis, by definition, represents how strong the reference genome is being covered by the sequenced fragments. Generally speaking, the depth of coverage is expressed as 1x, 2x, 3x, or more. Many researchers and scientists calculate the depth of coverage using the following formula. You would take the multiplication of the read length by the total number of reads and divide that by the size of the genome of the species that you are investigating. The second aspect of the coverage is the breadth of coverage, and it is depicted here on the x-axis. The breadth of coverage represents how much a reference genome is being covered by short reads. In other words, it represents the percentage of bases of a reference genome that are covered with a certain depth. On this slide here, I try to provide you with some of the recommendations that are based on experience and the literature in terms of sequencing data requirements for the different applications. So starting with the short read sequencing applications, if you are interested in de novo genome assembly using whole genome sequencing, you would need at least 100x coverage. If you are interested in detecting variants using whole genome sequencing or whole exome sequencing, then different data requirements are needed. For germline frequent variants, something between 20 to 50x might be enough. For somatic or rare variants, you might need to sequence at least 100x. And if you are comparing tumor tissues to normal tissues, then you might need at least 60x for the tumor tissue and 30x for the normal tissue. In case you decide to use short-read sequencing applications for population studies, then in this case, you might need something between 20 to 50x of coverage. On the other hand, for long-read sequencing applications, if you are interested in de novo genome assembly using whole genome sequencing, it is recommended to have at least 50x coverage. For gap filling and scaffolding and large structural variant detection with whole genome sequencing, it is recommended to have at least 10x of coverage. And finally, for germline or frequent variants, it is recommended to have something between 20 to 50x of coverage. Now that the sequencing has been done and we obtained the sequencing data, the next step in the workflow is data analysis and data interpretation. I'm not going to show you all the details of the data analysis pipelines, as these have been covered by previous webinars that you can check on our webpage. But just to give you a brief overview of the data analysis, in general, the data analysis pipeline is as follows. The first step is data QC. For this step, the input is the raw data that we obtained from the sequencing instrument, and the output will be clean data. So for this step, our interest is actually performing the quality controls to make sure that the data that we obtained from the sequencing instrument is of good quality. So there are several things that we can check for here, including the error rate, the GC content to make sure that it is not very high, and the sequencing quality. Once the clean data is obtained, we can proceed into the next step, which is alignment. For this step, the new sequencing data will be aligned to the reference genome of the species that we are studying, and SAM or BAM files will be generated. Using these SAM and BAM files, we will remove duplicates of sequencing data that might have been generated during the PCR amplification steps of library preparation, and thus they do not represent real DNA data. So we need to remove the duplicates in our data before proceeding. After the removal of the duplicates, we can perform variant calling using different pipelines and algorithms in order to obtain a VCF file. VCF is short for variant call format and is a file that could obtain that contains the information about all the variants that we can score for the new genome that we have sequenced in comparison to the reference genome that is available for the species that we are investigating. Once we obtain this file, we can proceed with further downstream analysis and data interpretation and focus on any aspect of data analysis that we might be interested in. And with this, I come to the last part of my presentation. So in the next 2 or 3 slides, I'm going to provide you with a brief summary and a short outlook about DNA sequencing. So to summarize the platforms that I have showed you today in terms of advantages and disadvantages, we know that Illumina provides us with the lowest cost in terms of DNA sequencing, has the highest confirmed output, and a very low error rate. However, Illumina has its disadvantages, and those mainly include short read length and limited de novo genome assembly capabilities. For long read sequencing and focusing on Pacific Biosciences or PacBio, the advantages here include real long reads, high accuracy with the CCS mode of sequencing, and for the first time, direct detection of epigenetic modifications. More importantly, all of this can be done with real-time data. However, sequencing with Pacific Biosciences is considered to be relatively high in terms of cost per GB, requires large amounts of starting material, and in general has high error rate with the CLR. And finally, for the Oxford Nanopore technology, we are now able to sequence real ultra-long reads that are even longer than PacBio. We can directly detect epigenetic modifications, and for the first time, we can sequence RNA directly omitting the cDNA generation step, and we can also detect RNA modifications. All of this can be done with real-time data output. In terms of disadvantages, the limitations of Oxford Nanopore include high overall error rate and large amounts of starting material that are needed for each experiment. Now, moving into the outlook and what we can expect for the future of DNA sequencing in general, I just wanted to take a step back here and look at the history of DNA sequencing from the last 20 years. So if you might already know this, but the first human genome was finished in the early 2000s, and that took around 10 years and came with a price tag of 300 million US dollars. Now, after 20 years of DNA sequencing, we are able to sequence the human genome for 1,000 US dollars within 24 hours. And this really puts it into perspective how much progress and developments and advancements have been happening in the field of DNA sequencing within the last 20 years. And based on this, we can say that the future is even brighter and more promising stuff is coming in the field of DNA sequencing. So there are a lot of research that is being done in order to expand the range of applications for DNA sequencing, especially for clinical diagnostic or translational research. Furthermore, different scientists and researchers are trying to develop better applications in order to accumulate challenging samples or to push the boundaries of the current methods in order to accumulate very little samples, such as single cells, or even by performing 3D spatial DNA sequencing. Not only the researchers and the scientists are trying to expand the portfolio and the advantages of the different applications, but also different sequencing technologies and the companies behind them are trying to do the same. For example, Illumina, one of the major sequencing technologies providers in the world, recently announced the NovaSeq X series, and with this instrument, their aim is to bring down the cost of sequencing the human genome down to 200 US dollars. In addition to that, and in particular, the short-read sequencing that is provided with Illumina, the company is also investing in long-read sequencing, and they recently announced the Illumina Complete long-read technology that would give us the advantage to sequence DNA up to 10 kb. And all of this can be done with the new enhanced chemistry called XLEAP XPS. And in addition to Illumina, other companies such as PacBio and Oxford Nanopore are also investing in the future of DNA sequencing. For example, PacBio is now investing in short-read sequencing, so they do not want to provide only long-read sequencing, but also short-read sequencing. They keep improving their workflows, and now they are trying to simplify the sample preparation protocols and even investing in single-cell long-read applications. Similarly, Oxford Nanopore technology is also investing in new chemistries that would improve the output of the sequencing platforms, are investing in single-cell long-read applications, and even more recently, the Oxford Nanopore sequencing technology has been shown to be compatible with protein sequencing. And with this, I come to the end of the presentation, and I thank you again for joining us today, and I'm happy to answer any questions you might have.
Thank you, Reid. Yes, we have certainly had some questions coming in as you've been talking, so I'm going to go through some of them with you now. So the first one we have is from Mark, and he's asking, what do you mean by a large amount of material being needed for third generation products? I guess, can you define what large amount means?
Yeah. I think this is best depicted in the table that I provided here. So if we take into consideration sequencing with Illumina for whole genome sequencing or whole exome sequencing, we need something between 200 up to 300 nanograms of DNA. However, if we want to do whole genome sequencing with PacBio or Oxford Nanopore, now the amount is several folds higher. We are talking about 5 up to 15 micrograms of DNA. So there is really a big difference in the amount of DNA that is needed between short-read sequencing and long-read sequencing methods.
Okay, lovely, thank you. So next question is from Galina asking, can you please give a more specific example on sample input limitations for Sanger sequencing?
Yes, for Sanger sequencing, the method is not very sensitive. So this is why there are certain restrictions in terms of the amount of DNA that is needed. So if there is a low amount of DNA, then we are not able to have enough Sanger sequencing, and the data that we will obtain in the end will not be of the best quality.
Okay, thank you. Next is from Ewelina. Apologies if I'm not pronouncing that correctly. Firstly, saying very interesting presentation, so thank you. The question is, could you explain a little more about DNA epigenetic modification detection by PacBio?
Yeah, so PacBio, with PacBio, since we are sequencing the DNA directly, omitting any PCR amplification steps, any epigenetic modification, such as DNA methylation, that happens on the DNA itself will be preserved. And with this, the instrument is actually able to determine that a nucleotide is methylated. I will go to one of the previous slides to explain it better. So if you look at this figure here, while the DNA is being sequenced by the polymerase, if we have, for example, DNA methylation at the cytosine residue, while the machine is measuring the fluorescence signal, there will be some kind of a lag in the fluorescence signal. And this is how the softwares and the algorithms of the instrument are able to define DNA methylation and determine that at this specific cytosine residue, there has been a DNA methylation footprint.
Lovely, thank you. So next is from Jose, asking if they are correct in understanding that exome enrichment only works with human or some model species. Is that correct?
Yeah, most of the whole exome sequencing panels have been developed to be compatible with mouse and human. However, there are some very similar species in terms of DNA that can still benefit from whole exome sequencing. And this would really require assessing how similar the genome is to the mouse or the human genome. And it is a matter of compatibility. At the end of the day, I believe that it can still be done, but there can be no guarantee in terms of the quality of the data or in terms of coverage if you are actually able to cover everything within the exome of the new species.
Okay, great, thank you. Next is a question from Rosa, asking how are target genes selected in TRS and what are the advantages of this compared to Sanger sequencing?
Yeah, so with target region sequencing, you can either already determine a set of genes that you think are relevant for a specific disease or a specific phenotype. So you can design a panel for more than 50 genes, for example, and say, okay, I'm interested in sequencing only these genes. Or you can say, okay, not only genes, but also genomic regions, such as the promoter or a regulatory element, such as an enhancer. In terms of advantages compared to Sanger sequencing, is that with target region sequencing, the sample requirements are less. So this is one of the main advantages. And in addition, we are actually providing data with higher accuracy since the coverage will be at least 500x for those genes or regions that we have decided to sequence. So the coverage is very high, and this would eliminate the possibility of any errors or mismatches that might have happened. And at the same time, we require less amounts compared to Sanger.
Great, thank you. Next is from Rowan, asking why is the accuracy of whole genome sequencing considered low, in inverted commas, comparatively, I assume?
Sorry, can you repeat the question? The last part.
So why is the accuracy of whole genome sequencing considered low? I would imagine comparatively, you know.
Yeah, okay, yeah, for this part here. Yeah, so at the end of the day, you have to imagine it like this. I can explain it in an example. If you have 1 million sequencing reads, if you decide to distribute them over the whole genome, then every part of the genome is covered only by a very small amount of reads. And then, of course, any mismatches that might have happened during library preparation or sequencing will not be eventually corrected. However, with whole exome sequencing, if now you concentrate these 1 million reads on 1% of the genome, then you are sequencing them more. And then, of course, you have more confidence and high accuracy in the data that you obtain. This is why the accuracy is better. And of course, it gets even better with target region sequencing, because now you are not sequencing 1% of the genome, you are sequencing maybe 0.01% of the genome. So the smaller the region that you cover with the same amount of data, the more accurate results you will get.
Yeah, great. Okay, thank you. From Mustafa next, asking, how does the gene selection process work with amplicon metagenomic sequencing?
Yeah, so we have usually a selected set of marker genes, such as the 16S, the 18S, or the ITS, that are common and shared between different microbes. And this is usually what we use in order to sequence and get information about all the different microbial species that might exist within the community. So we usually try to go for the highly conserved regions of the genome within the microbial species and sequence those within the using the targeted metagenomics approach.
Next up is a question from Bonnie, asking, can we use whole genome amplification methods to increase amounts of DNA for sequencing? And if so, do you have any method recommendation for this?
So if I understood the question correctly, what they are trying to do is to increase the DNA amount that they have with whole genome sequencing, or by...
Seems so.
It's not... Okay, if I understood it correctly, then they have a limited amount of DNA, and one way of increasing the amount of DNA that you have is by performing PCR. That would exponentially amplify the amount of DNA that you have. But my recommendation here is to be very careful with PCR amplifications, because it can introduce mismatches, and then it can introduce some defects into the pipeline. And that has to be tackled later during data analysis. So these are shown here in this pipeline. So these duplicates that can come while we are amplifying the DNA. And we definitely need to remove them, because they can create a bias when we do the data analysis.
Okay, thank you. Yeah. So I think maybe final question now. This is saying somebody is studying algae and considering long read sequencing. Would you recommend long read sequencing using Oxford Nanopore technology for this application?
Yeah. In fact, while long read sequencing has been very common nowadays for certain species like human and mouse, it still remains very tricky for some species, such as insects and aquatic organisms. And that can be because of how the DNA is organized within the nucleus in a complex secondary and tertiary structure. And that can affect the way third generation sequencing is performed, because if the DNA is folded in a certain way, that can actually block the nanopore proteins. And then, of course, the sequencing data that we obtain in the end will not be of very good quality. So if they are interested in studying aquatic species, I would really be careful about using Oxford Nanopore for long read generation sequencing.
Okay, great. Thank you for the answer. So I think that's all we have time for today. We hope you enjoyed the webinar and found it useful. Thank you for all of your questions. As mentioned at the beginning, if we haven't managed to answer yours live, we'll get back to you by email next week with the answer. In the meantime, we will email you a link to the webinar recording and the slide deck, along with a short survey that we'd appreciate you taking the time to complete. The survey will help ensure that we make future webinars even more relevant to you and your research goals. There will be a link in the follow-up email as well for you to view a certificate of attendance if you need one. So in the meantime, please stay in touch. If you have any upcoming NGS projects, now would be a great time to contact us about them, as we have an ongoing 30% discount promotion on a large range of our services. You can follow us on Twitter and LinkedIn, or get in touch via our website at novagene.com. So I would just like to give a very big thank you to Reid for a fantastic presentation today. and we look forward to seeing you all on the next webinar. Thank you.